Hello everyone. Today I'm going to discuss the GATE Zoology 2022 paper with you all. So let's get started. The paper, according to me, was easy to moderate because there were so many questions were directly from NCERT class 11th and 12th, and few were of UG level. So overall, the paper was from easy to moderate for zoology. So coming to the first question is which one of the following organic compounds is composed of only first the nitrogen containing base, second a single 5-carbon sugar, and third a triphosphate. So it's a question from class 12th NCERT and from the molecular biology chapter. You can go through it. And the answer to it would be nucleotide because it contains all of three, right? The only this is the structure of a nucleotide. As you can see, a phosphate group, a pentose sugar, and a nitrogenous base, right? A, T, G, C, adenine, thymine, guanine, cytosine, any of these, right? So the answer would be B, nucleotide. Coming to the next question is, which one of the following animals develops adaptive predator avoidance morphology because of the presence of high predator numbers in habitat? So basically, it is a type of adaptation. Fine. So adaptive behavior is basically adopted by an organism when that organism faces threat, right? So among these, the organism which exhibits this property is Daphnia, right? Now, talking about Daphnia, so Daphnia is basically a small planktonic crustacean, fine, which is commonly called as water flea because of its, saltatory swimming-like movement in water, which makes it, resemble like flea. So it is basically a planktonic crustacean, fine. And these species basically live in various aquatic environments like acidic swamps, freshwater, lakes, and ponds. Now Daphnia are basically, see, this is the picture of Daphnia, fine. And it is, very small, creature. So Daphnia species are known to show behavioral changes or modifications to their morphology in the presence of predator pheromones, that is chemical signals released by the predators. Fine. And sorry for the disturbance. Now, so basically, like the changes which they exhibit are like they, increase the size at hatching, they increase their bulkiness and the development of neck teeth. Like we have seen this bulkiness in case of birds also. Like whenever they, fear of any threat, they increase their size. So in the same way, this organism also, it exhibits morphological behavioral changes when it senses threat from its environment. So the correct answer would be A, that is Daphnia species. Coming to the next question, question number 87. To which class of Drosophila developmental genes do Fushi-Terazu belong? Right. So Fushi-Terazu, they are basically genes, right? So the question is, to which class of genes to this particular gene name it belongs? So the options given are gap genes, segment polarity genes, pair rule genes, and maternal effect genes. So if we talk about Fushi-Terazu gene, it is expressed at the blastoderm stage in 7 stripes that serve to define the even-numbered parasegments. So it is a pair rule segmentation gene that functions to define even-number parasegments in the early embryo. So since we are talking about pair rule segmentation over here, so it is basically a pair gene. Now coming to question number, okay, so the answer to this question would be C. Coming to the next question, question number 88, the action of which class of enzyme inhibitors can be reversed by adding an excess of substrate? That means to which enzyme inhibitor, which class of enzyme inhibitor, if we add excess of substrate, the enzyme would be inhibited. So it is uncompetitive inhibition, competitive inhibition, non-specific inhibition, or allosteric. So in allosteric, basically, the shape is changed by the inhibitor, for inhibition. Uncompetitive, we'll be discussing it. So in case of competitive inhibition, it can, if we increase the substrate concentration, it will promote the displacement of the inhibitor from the active site. Like suppose that this is the active site, right? And if we increase the substrate concentration, it will promote the displacement of the inhibitor from the active site. Okay, suppose that this is the active site and this inhibitor, it has binded to the active site. So if we'll increase the substrate concentration, it will lead to displacement of the inhibitor from the active site. So competitive inhibition can be completely reversed, right? If we add substrate so that it reaches to much higher concentration than that of the inhibitor. Fine. So it's basically like attaining the saturation. When the concentration of the substrate increases more than the inhibitor, so at that time, inhibitor will automatically get detached from the active site. So studies of the competitive inhibition have provided useful information about certain enzyme substrate complexes and the interactions of specific groups at the active sites. Correct answer would be competitive inhibition. Coming to the next question, question number 89. It is again a very easy question. If you have clear idea about, class 11th genetics chapter, NCRT, you will be able to solve it very easily. The question says, Mendel deduced the genetic principle of inheritance by experimenting on sweet pea plants. One of the experiments involved crossing plants with two contrasting characters, tall, dominant, and dwarf as recessive, which yielded all tall plants in the first generation. When the same genetic cross was independently repeated by a researcher, only short plants were obtained. Which one of the following can possibly explain the altered outcome? So the cross, as you can see, TT into TT, right? So this is homozygous, dominant homozygous condition. This is dominant heterozygous condition. This would be our first condition, which I have shown over here. So it will result into all tall plants, right? After crossing, how do we cross? This T into this T into this one, this into this, and this into this, right? So from this way, we got this thing, and we got the result in the first generation as all tall plants. Then we have the second condition, which is this one, like where we have heterozygous condition. This would be our second condition, which is the heterozygous condition. So again, when we cross it, we'll get it like this, like I've shown over here. So here we have 75% tall and 25% short plants. And then we have the third condition as this, in which we have one heterozygous dominant and the other is completely homozygous, right? If only these three conditions are possible, fine. Either we take heterozygous or we take homozygous, fine. And in the other case, either we take homozygous or we take heterozygous. So we have taken it all. This one, this one, this one, fine. So we have tested all the conditions. So in the third condition, as you can see, again, we'll get 50% tall and 50% short. So in neither of the conditions, we are getting 100% short plants. That means there is no such condition present in which we get 100% short plants. Fine. So since no condition satisfies the reason for the plants being dwarf, that means something is present which is, suppressing the dominant allele. This one, some gene is present, fine. So that some gene might be some suppression allele, which is basically suppressing the dominant, gene, right? Dominant allele. So that would be your, a suppressor for the tall allele. It is present in the dwarf. So the correct answer would be C, that a suppressor for the tall allele is present in the dwarf plant. So the answer to this question would be C. We can discuss the other options also. Tall plants are heterozygous. So we have tested that when we are taking tall plants as heterozygous, then also we are not getting any condition of 100% short plants. Second, an enhancer for tall allele is present in the dwarf plant. If an enhancer would be present, then certainly we would not get short plants at all, right? Because tall is already dominant. Leave the third condition. Coming to the fourth one, dwarf plants are homozygous. Though if we have taken dwarf plants as homozygous also, then also we are not getting 100% short plants. That means there is something which is, suppressing the tall allele. And that is the answer over here. I hope this explanation is clear. Coming to question number 90 to 92, and it is a multiple select question. That means more than one option would be correct. Which one of the following, question number 90, which one of the following is responsible for reversible receptor ligand interaction? So we have ionic, hydrogen, peptide, hydrogen interaction. Let me tell you, this structure you must be aware of, right? H2L2, right? So this is of basically sorry for the interruption. So as you can see, this is the structure of antigen, in which we have, these bands. So in this we have peptide bonds, dipeptide bonds, right? So peptide bonds are excluded over here. Since the term reversible is used, that means it can't be ionic also, because ionic is not, reversible. So we are left with hydrogen bonding and hydrophobic interactions. Only these two bonds, they are, responsible for the reversible receptor ligand. Apart from these, we have van der Waals forces also present in the receptor ligand interaction, because it is a reversible interaction. Basically, it is not a permanent or irreversible interaction. So the correct option would be B and D over here. Fine. Coming to question number 91, it is a very easy question. Like even an 8th standard student can solve this question. In the human body, which one of the following is involved in processing of a foreign antigen? So we have B cells, macrophages, RBCs, platelets. Like, it's a very easy question. So B cells, T cells, they are there involved in our immunity. Macrophages also, they form the line of defense. Fine. And RBCs, platelets involved in blood clotting. RBCs, they carry the hemoglobin. So over here, in the processing of foreign antigen, only two cells are involved, that is B cells and macrophages. Dendritic cells are also there, but here since only two are given, so the correct option would be A and B. Fine. Coming to the last question of the MSQ part, that is question number 92. Animals can be classified as specialist or generalist with respect to diet and habitat selection. Which of the following organisms belong to the specialist category? Now, let me tell you, specialists are basically those organisms which, eat only a particular type of food and they live only in a particular type of habitat. Fine. So they have these two conditions. That is, they have, niche for it. Fine. So they are known as specialist organisms. For example, you might have heard about pandas that they feed on bamboos only, right? Might have seen the polar bears. They live in the polar regions only, Arctic areas and Arctic areas only. Fine. The koala bears, they feed only on the eucalyptus trees. Since they have niche habitat, so they can't exist outside their niche habitat. So hence they are known as specialists. And generalists, they can survive anywhere. Like we have raccoon, or we can take any other organism which can survive in any other habitat. They don't require specific habitat for survival, right? So here the correct option would be B, C, and D: panda, polar bear, and koala bear. So I hope this question would be clear. This question can be solved by general common sense also. You don't need to have the knowledge. Uh, it is not necessary. You can solve it by general common sense also. Now coming to the question number 93 to 103. Here we have multiple choice questions, right, in which we have matched the following. The first is match the drug or chemicals listed in column 1 with the developmental physiological defects listed in column 2. So we have drugs over here, and we have the defects over here. So veratrum alkaloids. So first of all, whenever we solve such questions, we should see which one is the easiest one. So like here we have methyl mercury. It is the easiest one. And we know that it leads to Minamata syndrome. It's very easy. So first only we can separate out, okay, R ka 2 hoga, fine. So select the option in which we have we have R ka 2, fine. So we have this as option. Only one option we are having it. Then you can see for other options like thalidomide. Thalidomide, it leads to focomelia. Focomelia क्या है? Focomelia, it is a limb disorder जिसमें कि limbs develop नहीं होते अच्छे से. Fine. So Q का 4 हो गया. Fine. Then we have diethylstilbestrol. Diethylstilbestrol, it will be related to obesity. Fine. So we have S1, and then at last we have veratrum alkaloids, in which will we have cyclopia. So cyclopia is basically a genetic disorder. So the correct option would be A, 93A. You can see the explanation here. Veratrum alkaloid, cyclopia, thalidomide drug, focomelia, diethylbestrol, weight gain, obesity, methyl mercury, Minamata disease. Fine. Coming to the next question, number 94, is we have these organisms, and most of them, they belong to the phylum Platyhelminthes, the parasitic phylum. So here we have the first one, Asquirus Lumbricoidus. Fine, that structure you might have studied in class 11th NCERT, such structure. Fine, so it is present in the small intestine. So it's the P3. Then we have Ranunculus mediensis. It lives in the subcutaneous part. Then we have Enterobius vermicularis. It lives in the cecum or vermiform appendix. Fine. And last we have Bichuria bancrofti, which lives in the lymphatic vessel or lymph nodes. And it basically causes the elephantiasis disease, Bichuria bancrofti disease. Fine. So the correct option would be D. It's again a very easy question. If you have studied class 11th NCERT, chapter number, I guess, 1 or 2, so you would easily solve this question. Till now, I found no such question which goes beyond the NCERT of 11th or 12th. Till now, I haven't found any question till question number 94. Maybe we can find ahead. Now, question number 95, we have to match these are the sources. Column 2, match cell types. We have cell types, then we have their sources, and then we have the function roles, the primary function roles. So in this also, we should target the easiest one. Leydig cells. We all know Leydig cells, they are present in the testes. Right?